we start the webcast. Look at my sandals today.
Yeah, I saw those.
They're sporty though. We're
I'm a little
good.
jealous, actually.
I gotta be. I was like,
Otherwise you won't make it to the end.
I know. I hardly made it to the end of it.
Got your deck shoes.
Okay. I do. I was like, man,
I'm a little
my
jealous.
shoes are good on a normal day. It is not a normal day. I need some flip flops.
I don't like being 5'2".
I know that's my problem. So now I'm really. I don't. Now I'm really my own.
You're what? You're really running? I really am not. I am 5'4".
Jeff, you ready? Good. Thank you all for coming back. Right now, I'd like to introduce Pam Vig with Mirum Pharmaceuticals, and she is going to talk about clinical trials and drug development. Thank you, Pam.
Thank you. Okay, let's see if I can. So, my name is Pam Vig. I'm the Chief Scientific Officer of Mirum Pharmaceuticals, and as Roberta said, I'm going to talk about clinical trials, what Mirum is up to, who we are, and just kind of introduce how we work. But first, I really want to say thank you to Roberta and Cher for everything, and I think we all can agree that this has been a pretty phenomenal meeting. So thank you very much for that. And I also wanted to start by saying, when we do clinical research studies in industry, we spend a lot of time talking to a lot of different people. We talk to basic scientists, we talk to the disease experts, we talk to the FDA, we go to conferences, we just keep going through all of the research and keeping up with it and trying to make sure that we design our clinical trials appropriately. But what we don't get that often is this. And so this has been pretty phenomenal experience, and everyone's been so open. It's been incredibly appreciated and very humbling and also very motivating. So there's a few of these characters up here in the front. We have Mike Gray, if he can raise his hand. There's Ian Clements from Mirum. We have Cindy Cochran right here, and Chris Peets, who's our CEO, and there is Bernard, head of commercial. So this is Mirum. We are really a company focused on pediatric cholestatic liver disease, and the company is really built around working in Alagille and PFIC. And so who we are, we're formed very recently, November 2018. We're based in San Francisco and in Basel, so we sound very international, but we're really like 25, 26 people. And just a little bit of history. So you, although we're a new company, you have probably heard of LUM-001, or what's referred to as a Lumina drug, or the Shire drug. And so this compound is called maralixabat, and it's really come full circle. It started for Alagille syndrome and for PFIC with Lumina, in which Mike Gray was the founder, and has really been years in the making and worked its way from Lumina to Shire and now back to Mirum. And so our intent is to really get this drug out into the hands of the treating physicians so that there are other treatment options. So what do we do with clinical research? So with everything in life, and certainly being here the last few days and listening to the group and the different perspective of everybody, it really is a collaborative effort. And it involves us, it involves our clinical trials, which are really underpinned by the scientific efforts. And for example, Nancy Spinner talking about all of the genetics, and thank you very much for that talk. That was great, Nancy. And that really drives our energy, our efforts. We pick up on the things that we need to understand in order to design our clinical trials in a way that is meaningful for you and for the FDA, because at the end of the day, the FDA has to check their boxes. They have a job to do. They need to make sure the drug is safe, it's effective before they approve it. This involves study sites. So clinical research studies, the amount of work that goes into each study from the center involves the treating physician, the study nurses, the coordinators, the folks who draw the lab, the people who put the contracts together. It's an army of people, and it is standardized from center to center at every center that you work in around the globe. And then at the center of all of this is the families, and this Alagille Alliance is really the center point, I think, for what we're trying to accomplish. And it's an unbelievable community, and the voice that you have is very important in how we direct our studies and our efforts. So from our perspective, we have three main groups that we need to really pay attention to. One is the physician perspective. So we spend years talking to the physicians. And this goes through your phase 1, your phase 2, and your phase 3 studies. And your phase 3 study is what you do right before you ask the FDA for approval normally. So as the field changes and it moves on, we continue to hone in and focus our studies appropriately based on the science, based on the clinical research, based on the patients. So we spend a lot of time talking to physicians. We have spent a lot of time talking to the patients because what we are doing is using a patient-reported outcome, and I'll tell you a little bit about what that means on the next slide. And so there's been a lot of years spent, starting with Lumina, of gathering data, family surveys, questionnaires, trying to understand how things are measured, because these are not labs, these are not physician-reported, these are reported by you. And so sorry, something just flew in the air. What was that? So, yeah, as long as it wasn't this way. So we've had patient-reported outcomes. So I just want to say that all of the data registries and all of the interviews that are done and all of the questions that are had and all the surveys you fill out, they maybe feel like you complete them and they go into a black hole, but they don't. They don't go to waste. They are used as information that's incredibly important, and so it's much appreciated. The FDA perspective. So they are divided into divisions. And so for this compound, which really targets the liver for Alagille, this is with the GI division. And so within the GI division, there are hundreds of diseases that they have to look at. So it's really our job to make sure that we're providing enough information. So they bring their own disease experts to the meetings, into the discussion, into the documents, to understand the disease and to decide what endpoints we need to use in order to satisfy their criteria to get the drug approved. And at the center, again, of all of this is the patient and the family voice. And I think Mike said it last night, that really advocating and talking and speaking up is really important. And I was really sad to see on the survey that somebody had said that when they completed the survey for the Alagille Alliance, that was the first time they felt that they had a voice. And so I think this is a great place for you to put your voice. It's kind of the center of what I think everybody is trying to accomplish for Alagille. So please keep doing that. So what is your role in clinical research? The information you share matters, as I said, the interviews, the registries, the conversations. It helps us advance our understanding of Alagille syndrome. It helps us design our clinical trials. It helps shape the design of the studies. The burden of the questionnaires that I appreciate. There's a lot of questionnaires that we ask, and many of you are on clinical trials. These are the instruments that we need in order to continue to move it forward. So it provides a platform for the FDA discussions. And I put these 3 last bullet points in here about participation in clinical trials, be aware, be informed, to consult with your doctor. Now I feel like I need to change those because I heard yesterday that, you consult with your doctor and oftentimes they don't have the information that you need. And so you're looking and you're Googling and you're on the website and you're doing your own research and there's really nobody to go to. So I think we need to be better at that and making sure that information is disseminated in the appropriate way. Okay, so a little bit about patient-reported outcomes. So these are not measured by the lab, not measured by your physician, although that is all supportive information to what we're trying to do, but really because what we're measuring is itch. We're measuring the pruritus, and so that's what our compound is focused on, the cholestasis part of Alagille, so increased serum bile acids, pruritus. How it affects sleep, how tired your child is, what's the quality of life, and other measures of how you and your child are doing. And so this has been developed through a series of interviews over years with families. It's been validated work, and I saw on Nancy's, one of her last slides, it said validate, validate, validate. And so that's one of the things that we have had to do in working with the disease experts and the FDAs, and it's been translated into many, many languages so that things are consistent when we run a global clinical trial. That when you see a question of how severe is the pruritus, what's the frequency, that's translatable in every language and the meaning is the same. So that requires quite a bit of work. Okay, so a little bit that you guys are all very familiar with this, but essentially cholestasis in Alagille and in other cholestatic liver diseases is basically an impairment of bile flow and a reduction of bile in the intestine, and so there's a retention of bile in the liver that is toxic, is damaging. You get elevated serum bile acids and associated pruritus and other things along with that. And so by reducing cholestasis, it reduces itch, it reduces serum bile acids, and biliary diversion addresses some of this. As we heard from Dr. Kamath, it really works in only about half of the patients. But it can improve liver enzymes, the itching and the associated, there's associated complications that come with it though. And sometimes, and you guys know firsthand that you have to make a decision not knowing if it's gonna work or not. And so non-surgical therapeutic options to reduce the bile acids and the cholestasis is really what we're trying to accomplish. So maralixibat, or LUM-001, but now we call it maralixibat, is a apical sodium-dependent bile acid transporter. And really what that means is it interrupts the bile acid recirculation. So bile acids are retained. They are about 95% recycled, and what this compound does is it interrupts the recirculation of the bile acids. Oop, I have a pointer. Hold on. Okay, so essentially what the compound does is instead of it going back up to the liver, it comes out of the colon and thereby reducing and lowering the systemic levels of bile acids in the blood and bile acids in the liver, thereby depleting the pool and having an impact on pruritus and cholestasis. So our recent data, I'm going to walk you through our most promising study, and we have gone to the FDA with this trial, and Roberta came with us and was incredibly impactful in her voice and representing herself as a mother and representing this group as well as a Alagille Alliance president. So this study, I'll walk you through the design and the results that we've seen, and then what our plans are. So this is a 400 microgram per kilogram per day dose, so small. This compound is not systemically absorbed, which means it's only in the gut, so you don't have a systemic absorption and the side effects that come along with it being in the blood. It is an oral solution, so it's given, it's grape flavor, so it's given like a children's Tylenol. And so all patients received maralixibat for 18 weeks. So everybody's on drug for 18 weeks. And then at the time of week 18, you have half of the patients that stay on maralixibat, half go on placebo, and patients and study participants don't know what they're going to be going on, or didn't know what they were going to be going on. And placebo is really just it's as if you're taking the compound, but there's nothing in it. So it's like taking Tylenol, but there's no active ingredient in it. It's just a grape-flavored solution, and so there's no drug on board. And this is a way to really weed out what the treatment effect is, to see what happens when you're on drug, what happens when you take drug away, and then what happens when you put study participants back on drug, because this is a patient-reported outcome, and their measurement has to be done in such a way because it's not objective. Okay, I've got the wrong one. Hold on. I'll figure this out. Okay. So the study is called ICONIC. Methodology is really the way that we study our clinical protocols, and in that we have to be, we have inclusion and exclusion criteria. So this is where we say, these are what the serum bile acids need to be, these are what the liver transaminases need to be, this is the age of the subject, these are the things that they can't have to be included in the protocol. And that's so that we can study a group in where there are fewer variables, and in that way we can say that there is a real treatment effect, rather than saying, well, that's because you had a whole bunch of different patients and a whole bunch of different variables, so we don't really know what's going on. So this is a way to really, it's an experiment, it's a study, and it's the way that things have to be done for the FDA to feel that the result that they're seeing is real. So these were kids age 1 to 18, and I know that there's a few, not from this study, but from other studies, a few children that have been on maralixibat over the last few years in this room. These guys have chronic cholestasis, so they have high serum bile acids, they have significant pruritus or itch, and the measurement is called, for us, is called the itch row, which is itch reported outcome by the observer, and the observer is the caregiver. It's the parent, it's you. And these children did not have biliary diversion surgery or liver transplant, so they basically had high serum bile acids and they had pruritus. And we looked at endpoints. Primary is are the thing that we're looking at is pruritus, and then everything else is supportive. So the clinician scratch scale, where you go in and the physician looks at the skin and makes a decision and a scoring on what they're seeing and see if it's supportive of what you were seeing. And then looking at other objective measurements like serum bile acids, et cetera, and we also looked at xanthomas. Okay, so what did we see? So this is the demographics. This is a usual slide that you see on who were those study participants. What did they look like? And there were 31 of them, age about 5.5 years. Everyone had the JAG1 gene. And the itch row score, so this is a score that's 0 to 4, 4 being very severe for itch, 3 being severe, and then 0 being none. And so you can see that these kids came in pretty itchy at about 3, so severe pruritus. The clinician scratch score is a 0 to 4 scale, and again, this is showing what we see here. And then high serum bile acids. Okay, so this is the first slide on the results. So this is looking at serum bile acids, and we see, I'll just explain these lines. So you have, in blue, you have MRX is maralixibat. So maralixibat. That means they received maralixibat from week 0 to 18. And then this 4-week period that's here in pink is when the children were randomized to either placebo or to stay on drug. And you can see that during this time, the kids who received no drug, their serum bile acids spiked back up to where they were at baseline, and then when drug was reinstated, it came back down. And this was mimicked by what we saw with itch, so pruritus, based on that itch row measurement tool that we talked about. Okay, Roberta's flashing a 10 at me. I think that means I have 10 minutes left. I'll be quick. So we see all patients coming down. So this is a pruritus measurement, and we see that they're coming in with a score of 3, and there's a pretty significant reduction in their itch. And the kids that received no drug during the 4-week, or received placebo during the 4-week window of randomized control, their pruritus came back pretty significantly and then reduced again when they went back on drug. And all of this is in line with which makes sense because it's what we saw with the serum bile acid results as well. And this is just showing kind of what happens in that 4-week treatment period and how the two separate on a bigger scale. This is what we call a spaghetti plot. This shows every single subject, every single study participant that's on the study. These are the guys that were on maralixibat, placebo maralixibat. The study participants had a significant reduction. As you can see, everybody went back up here and then came back down. And here are those that stayed on maralixibat, and those that stayed in response, had a response, stayed in response, and those that didn't have a response never had a response. So this doesn't work in everybody, but where it works, it seems to work well. This is corresponded. So a lot of the kids, if they were old enough to fill out the questionnaire themselves, it was filled out by the caregiver, and it was also filled out by themselves with the help of a parent or caregiver. And the results that we saw were very similar, where same thing happened, and it mimics what we saw back here. And then the clinician scratch scale. So this is a scale that was when the study participant would come in, and 4 being the worst of cutaneous mutilation or hemorrhage, and scarring, so pretty bad pruritus and itching. 3 being that abrasions are evident, and then all the way down to 0. And you can see that this is a baseline level. And in black and in this dark gray, these are scores of 4 and 3. So again, showing the severity of the pruritus at baseline. And at week 18 and week 48, which is when everybody was on drug, you see that most of the study participants are at 2 or below. This is during the 4-week treatment period where there was a randomized withdrawal, and this is the placebo group here where you see that during that 4-week treatment period, they again went back to scores of 4 and 3 mostly. Cholesterol is lowered with this drug. It started off actually as a cholesterol-lowering drug way back when in adults, and so we know that that's an on-target effect. And in that, we saw a reduction in xanthomas in these kids. So about a 44% reduction was observed in xanthomas, in line with supporting the reduction of serum bile acids and what would be mechanistically would make sense. So one of the questions that you had in the survey was also, would you be willing to participate in a clinical trial if there were a lot of side effects? And so that was really a great survey for us to see how people feel about that. And so this is what is normally shown with regard to adverse events when we present our data. So this is sort of AEs regardless of relatedness, and that means that whether or not it was related to study drug is immaterial. Everything is recorded on that data sheet. And so the side effects of maralixibat are really GI targeted. So because the bile acids are being depleted out of the colon, there's a diarrhea associated with it. There is some abdominal cramping and some discomfort, but it's been shown to be transient in nature, and it's mild to moderate, and after the gut gets accustomed, it seems to go away. And we have kids that are on maralixibat for over 4 years now, and have a maintenance of durable effect, maintenance of decreased serum bile acids, maintenance of pruritus reduction, and they no longer have the diarrhea. So these are sort of everything I talked about. Just the conclusions is that, there was a significant reduction in itch and serum bile acids over time, improvements in quality of life associated with that. The AEs were mild or moderate in severity, and they didn't last for the GI, and we continue to monitor very closely all of the safety because this is very important for the FDA in order for them to make their decision and make sure that what they're approving is okay. So what are we doing next? So what's happening next is we've had regulatory discussions with the FDA very recently, just a couple of months ago, or maybe a month ago, and the European Medicines Agency, which is like the FDA for Europe. We're trying to file on our phase 2 data, so on this existing data. This is a phase 2 study, not a phase 3 study. And because the results are convincing, and because we have long-term treatment with these patients, half of the patients remained on study, that we feel that because there are only surgical therapies associated with reduction of serum bile acids that really seem to work, that a non-surgical treatment option that where if it doesn't work, you can just stop it, is really important. And so we've had discussions with them. They were very open. They may ask us to do a phase 3 study. They may be willing to accept this. We don't know yet. In any case, we will be conducting a clinical trial in 2020 either way, because even if they do say, sure, you can go ahead and file, that process is quite long. So we'll have something for you. And then lastly, because we're in the fortunate position where we have patients on maralixibat for several years, we are in a position to compare the long-term benefits with the natural history. So we're working with Benita Kamath. She gave a nice talk yesterday on what she's doing with GALA. There's a lot of patient data in that registry. So it really helps us further our understanding of what happens when you reduce serum bile acids over a period of time, and what might be the benefits or not of that. And so lastly, I think that's it. I just really wanted to say thank you so much for the opportunity. We are really delighted to be here, and it's been a really important conference for us, and quite humbling. So thank you.
Thank you, Pam. So before we go to the next